So if you forget my presenter note, what you see over here is the first cell division of the zebrafish embryo. So this will continue going on through 8 rounds of cell division until it undergoes gastrulation, and then you form a whole zebrafish. But for the purpose of this talk, we are only interested in the first cell division. So not much is happening, and it's a pretty simple system, and that's why we like it. Okay, so let's just take a step back, and then I'll explain what the usual cell division looks like. So this is kind of like your normal cell, as you probably seen many times in biological textbooks. So this is a cell. It divides into 2 nucleus, and then at the region where the microtubule from the 2 nucleus overlap, it's kind of forms a zone of enhanced contractility. And then this cable, this furrow then contracts, and then it basically pinches off, and then you get 2 cells. And then this is what this video is showing over here. However, in zebrafish, we have a hemispheric cell that is sitting on a massive yolk. And then the way this cell division works is that you still have the 2 nucleus, and then you still have the microtubules overlapping, and then you get the zone of enhanced contractility. And then this contracts, and then it sort of divides down like this, a bit like cutting butter, and then not like tightening a belt in a sack like this. And then this is what this video is trying to see. You can see the first cell division and second, and etc. So the biological question we have is to understand how this partial cell division is achieved. And then to do that, we will take a step back and look at the structure of the cell. So this is a nucleus, and then this is the cytoplasm, which is sort of like where the microtubules are, and then you have some loose actins. But then if you zoom in at the surface of the cell, so firstly we have this membrane, which is sort of a really soft layer made of lipid. And this is very deformable, and then it's very fluid, and the physics of this is actually very interesting, but we will not go into this in this talk. What we're interested in is the cell cortex that is just below the membrane. So this is a layer with many different types of proteins, and then it's kind of like a stiff shell that holds up the shape of the cell. So we think this is the part of the cell that is important to us in deciding what the shape of the cell is gonna be. And then in particular, in this large group of proteins, we have actin fibers, which are just like long chain polymers like this. And then you have the myosin motors that attach both of its ends to different actin fibers. And then by this one walking this way, and then this one walking that way, it's able to exert contraction forces on the actin fibers. And so to kind of look into the dynamics of the cortex, we team up with our collaborators, Xintong, who is a PhD student of C.P. Heisenberg, also at ISTA. And then they were able to image basically, this is an actin label, so they were able to image the actin density in the cortex. As you can see, this thin layer is the cortex that I just described, and you can see that the actin density is just a very thin layer under the cell membrane. So this is what it looks like at this magnification. If we zoom in a little bit, then you can see that this is where the cable is going to form. So first you can see is that there are actually 2 kind of slightly different types of actins. We have the aligned actin, which forms the cable, and then we also have the punctate actin, which doesn't have any particular alignment. Should I pause or should I continue? Yes. Okay, it depends on, okay, if I turn it this way, I'm a little bit louder. Okay, so there are 2 types of actin. One is this type of aligned actin, which is where the cable is, and then the other type is a punctate actin. You can see there's no kind of specific orientation or order, and then they're just kind of like dots of actin that are randomly oriented in the cortex. And we can see this a little bit more clearly if we look at the snapshots. So to describe the alignment order of the actin, I'm using nematic order, and I'm sure I don't have to explain what this is. If you're not already familiar, it's just kind of a headless vector that tells you how aligned the actin fibers are. So if it's isotropic, which means the orientations are entirely random, then the nematic order will be 0. And then here we look at the QYY component, which means this is positive if the actins are aligned in the vertical direction. So here, by processing snapshots of these images, we can see that this is before this actin cable is formed. We can see that the orientations are basically random, and then we have some sort of, if you look very carefully over here, you can see there's some small inward velocity towards the band, towards this cable as the cable forms. This is also shown in the compression over here. You can see that in the middle where the cable is about to form, there is large positive compression. But then in the second phase, after this cable is formed, is what's super interesting. So here you could already see it very clearly from the video. We actually have a outward velocity over here that is flowing away from the band. And this is particularly weird. And then our kind of what we were super curious about is how does this outward velocity form, and then why does it form? Is there any biological function to this thing? Okay, and then to do that, we decided to use active gel theory to describe the actin cortex. So active gel theory, as we heard about plenty in the first day, it's a mesoscopic description of actin filaments as soft, overdamped materials. So by mesoscopic, I just mean that we will kind of look at a length scale that's much larger than a single filament. So we will not be looking at the details of the treadmilling of the myosin fibers on the actin filaments. And then I'm sure we all know what soft is and overdamped is, so I won't explain. So there are two ways to arrive at the theory. So the first way, we could use a top-down construction. So we look at the conservation laws that we think is in the system, which in this case is on the short time scale, kind of on the time scale that's much slower, that's much faster than the actin turnover rate. We assume mass conservation. And then obviously there's no momentum conservation because it's an overdamped system. And then we also think about the symmetries we have in the system, which is importantly like rotational symmetry, rotational symmetry and translational symmetry, and also importantly the lack of time reversal symmetry, because this contraction by the myosin walkers is highly active and highly non-equilibrium. So, and then we could take the lowest order terms in the density and the gradients, and this gives a theory. So this approach has been, there's a huge body of literature on this, and these are only 3 of the citations. So, so it's been shown to work pretty well for the actin cortex. But obviously, another approach you could do is the bottom-up construction. So you take the microscopic components, and then such as the actin fibers, the myosins, the cross-linkers, or even the membrane, and then you write down all the interactions you think is happening between them, and then you do an explicit coarse graining that arrive at this like mesoscopic description. So this has also been done in this paper, and then the result is that it sort of gives you the similar type of terms. So for our talk, we'll follow this approach. So this is so I will just be going through the equations of the active gel theory one by one, and then we'll try to understand what this is about. So if we look at the density dynamics, so here rho is the density of actin. So the density changes by advection, and then the actin turnover, which is the polymerization and then depolymerization. But at the end of the day, what this term does is that it pulls the actin density towards a certain target density. And then lastly, we have diffusion, which is just needed if you don't want your simulations to blow up. And then we have the force balance equation, which is say friction is equal to the differences in stress. And then lastly, we need the constitutive equation, which relates the stress to the other components, to the other variables in the system. So the first term is just the classical Navier-Stokes term, which is the stress is proportional to strain. And then the second one, we have a bulk viscosity, because what we've done is we are going to look at this actin as a 2D theory, whereas in reality, this cortex has a little bit of thickness. So even though it might be incompressible in 3D, once you integrate over the thickness, the theory becomes compressible. So that's why we have a little bit of bulk viscosity. It actually turns out to be not very important. We just put it here just for completeness. And then the third one is, so the simplest assumption is that regardless of the alignment of the actin fiber, they contract isotropically in all directions, as shown over here. And then this is driven by the myosin motors. So obviously this term is the only active term in this equation so far. And then this is the term that breaks detail balance and etc. So this is the equations for the density field and the velocity field. But then to look at the formation of the aligned actin cable in the middle of the cortex, we also need to look at nematic order. So this is sort of what I said, but just a little bit of a revision diagram. And so if we look at the dynamics of the nematic terms, so first we have the material derivatives of the nematic order is equal to some sort of relaxation towards the ground state. So this is just saying the nematic order would like to be 0 if nothing else is happening. And then also it would like to be aligned with the neighbors. So this is the usual passive nematic term that you would put in. And then this one is actually also a passive effect. This just says, if you imagine kind of like a soft squishy ball in a flow field that is like this, then you can see that the flow field would squeeze this ball in this direction and this direction while pulling in this direction and this direction. So naturally it would form it into a rod that's aligned in this direction. And then that's what this term is saying, is just saying that the nematic term is under the squeezing of the local flow field. And it turns out that if you just do all of that, then it is enough to explain the cell division in C. elegans, I believe. So here, this is a paper in 2016 where they applied this theory I just described, that is entirely isotropic, to the nematic order field observed in the division of C. elegans. So what we have is that we have a contractile zone in the middle, which pulls the actin fibers from both directions, which forms a velocity gradient. And then this velocity gradient, by this kind of nematic and velocity coupling term, then squeezes the actin to be sort of aligned in this direction. Does that make sense? Okay, so I mean this is quite important. So I guess I'll try to explain a little bit more clearly. So basically you have the contractile band in the middle, and then you pull actin density from both sides, and then this velocity field that's created, kind of like shear-ish velocity field that's created, is then squeezing the actin fibers in the middle into this sort of like a band shape, into the alignment order that's in this direction. Okay, so then we ask the question, okay, before we add bells and whistles and all the extra things that we think is happening in our biological system, is this simple theory that has been shown to work for complete cytokinesis work in our cases? So it sort of does, as you can see that after a little bit of luck and parameter guessing, we arrived at a snapshot that, and you can see that, so this is density, this is velocity, this is the compression, and then this is the alignment vector. So you can see that, in fact, by just putting a zone of enhanced contractility in the middle, we were able to make a nematic order alignment like this. And then if you look at the velocity field, which is, you can see a little bit more clearly in the time evolution, in this like time evolution plot, where you have blue at the start and then you have yellow at the end of the simulations. You can see that we do get a little bit of negative velocity over here. That is, we do get a little bit of positive velocity going to the right and negative velocity going to the left, which corresponds to this little arrows over here. So we do have a little bit of kind of outward flow. And then we do have a little bit of this like sort of, we call this depletion zone, where we have a kind of reduced density on either side of the band, which is another thing we observe in experiments. So the way this happens is that the actin contraction in the band leads to this depletion zone because it's sort of attracting all the neighboring actins, and which means you have a region of lower density in the depletion zone and then a region of higher density in the rest of the cortex, which means the cortex is then going to attract more actin from here because there's more density, because the contraction is proportional to the density of actin. And that's the thing that's creating this tiny amount of outward flow from the depletion zone to the rest of the cortex. However, this contradicts our experiments. So the first experiment you see over here is a laser cut experiment. So this is I've been told this is very annoying to do. It's just you take a laser and then you cut a little bit of the cortex and you see how it recoils, and it gives you information about the amount of contraction forces at this particular region in space. So here what I'm showing is that to get this particular velocity pattern, what we need is very small hydrodynamic length. So you could think about hydrodynamic length as the length scale over which the density profile or the velocity profile varies. So in this case, the hydrodynamic length is about this much. So it's sort of like the decay length of, it's kind of like the length of variation of this. And you can see that for this to work, we need hydrodynamic length of this much. Whereas the laser cut experiments, if the hydrodynamic length is extremely small, then we wouldn't see sort of this long range reaction to the laser cut. We would only see kind of like a very short range contraction. So in conclusion, what this experiment is telling us is that the hydrodynamic length in our system is pretty long. As you can see, you cut here and then immediately the actin here is started contracting. Whereas in our simulations, to get this, we require small hydrodynamic lengths. So that's clearly on conflict. And then another thing is that if you see over here, then actually very close to the band, we have sort of isotropic flow field. We have the velocity going in over here, in over here, in over here, and over there. So that means for this particular regime in simulations, we require kind of isotropic contractions everywhere. And this again also contradicts with our experiments. So we can see this is the contractile band. And then if we look at the velocity field around the edge of the contractile band, we can see that it's going inward at the end, but it's clearly going outward with no like inward velocity on the lateral side of the cable. Okay, so then we started thinking, okay, let's see what ingredients we should be adding to our theory to make this able to describe our experiment. So the first thing we came up with is, okay, then I mean clearly the way the myosin works, then maybe we need to add some nematic contraction, which says which basically says if the actin fibers are aligned this way, then it's going to contract in this direction a lot stronger than compared to this direction. And you can see how kind of intuitively this would give rise to the observed flow patterns, right? Because our flow pattern is basically goes inward this way and then it goes outward that way. However, this turns out to be not enough because the flow generated by the nematic order, which is like that, is actually destabilizing for the nematic order from the velocity from basically this then would squeeze the nematic order in this direction, which will cause it to want to align in entirely the opposite direction. So having nematic contraction and the flow alignment term is entirely it acts in entirely opposite ways. So this doesn't work. And then we're like, okay, what about another thing we know in this kind of systems is that there is a little bit of catch bond mechanism where once the actins are aligned in a certain direction, then it's happy to stay this way, and it's actually stronger in the direction that it is aligned. So effectively, mathematically, this basically just means there is some stabilization mechanism for the nematic order. And we also put that in, which almost works, but it's incredibly difficult to obtain both the depletion zone and this enhanced density in the actin cable, as well as this flow pattern. So you can see that via a tiny change in the parameters, we suddenly go from this thing where we have a band in the middle and no depletion zone to no band at all, and then just a depletion zone. So we also didn't like that either. And then for the last step, we actually resisted this for a long time. We tried a bunch of other things that I'm not showing today because we were really trying to be like, okay, we're statistical physicists, right? Are we really going to make our model this complicated? But at the end of the day, we're like, okay, this is what the biologists tell us. We have to do what needs to be done. So here is a labeling of myosin, so which is the motors I tell you. So you could kind of think about myosin as the myosin label as indicating the regions where the actin is able to contract actively. So you can see that even despite all these outward flow field, the myosin is still quite localized just in the actin band, and it doesn't seem to be going into the cortex at all. And then this inspired us to think, okay, maybe there is some difference in material properties of the actin that's in the cable and then the actin that is in the cortex. So what we should be doing is to model them as two different type of actins. We'll model the actin that's in the cortex as passive actins that doesn't exert contraction, and then we'll model the actin that is in the cable as contractile actins that's able to exert contraction. And then we say that they're both connected to a big bus of monomers, so they can exchange densities between each other. So this is what I call the two-component active gel theory. So this is all the equations of the two-component active gel theory. It's actually very similar to before. The first two equations is this one tells you, this is exactly the same as before. It's just here we are just looking at the evolution of the density of the active actin, and then here for the passive. And then here, this is the constitutive equation. So rather than putting just the active isotropic stress, we also putting the active pneumatic stress over here. And then this is just a term that stabilizes the pneumatic order. And then for simplicity, we just put it as a spontaneous phase separation. So if it's kicked in one direction, it would then tend to form pneumatic order alignment in that same direction. So with this, we scan the parameters in the pneumatic contractility and the isotropic contractility. Oh, okay, nice. Okay, we scan the parameter regimes in pneumatic contractility and isotropic contractility. And then broadly speaking, there are sort of three different types of patterns that we get in the system. So let's look at the C region, which is the first, which is high isotropic contractility and then low pneumatic contractility. So this is just the same as the C elegans example, where we get isotropic contractions everywhere, but then just because of this squeezing effect by the velocity field, we get a pneumatic alignment. We get a pneumatic alignment in the cable. And then we have A, which is most similar to what we have in our experiment. So this is, we have the formation of the cable, we have the depletion zone, and then we have a velocity field that is kind of going inward over here. And then this is the outward flow that we've been talking about over there. And then again, we have the nice pneumatic order alignment over there. And then there's also this weird behavior, which is in the B region. And this happens when the pneumatic contractility is extremely high. And the system somehow finds another very stable state where the alignment, rather than vertically, they're all aligned in the horizontal direction. And you can see that this is also stable because they're aligned in some direction, they're happy, and then also and then this then draws in velocity field in this direction and going out in that direction. So this is not we observing experiments, but it was just kind of a curiosity that it is part of our phase diagram. Okay, so for our experiment, obviously we take the A region, which looks a lot like what we have. And then you can see that if we look at the chemograph of the time evolution, so this is taking a line over here and then looking at the time evolution of the densities on this line. So you can see that in simulations, we have firstly the formation of the actin cable, which is this bit. And then you can see that we have the inward velocity over here, as we see in experiments over there. And then we have the outward velocity over here, as we see in experiments, which then also form this depletion zone that slowly gets larger over time. So we were pretty happy with the agreement with the experiment. And then they asked, okay, what are the other predictions or sort of things that are consistent with our active gel theory? I'm a bit hesitant to call it predictions because they did the experiments first. Okay. But in the talk you always have to say, oh, this is what our theory predicts. But yes, so with that grain of salt, then the predictions of the active gel theory is firstly the properties of actin in this aligned band. is very different from the properties of actin in the rest of the cortex. So, which, as you can see from the laser cut experiment over here, the actin in the band is extremely contractile over there, whereas if you cut the cortex, basically nothing is happening. There is no contraction that we can measure. This is the tiny laser cut you see over here, and there's basically no recoil from this. Another thing is that we predict that since we are in this region of the phase space, we have high nematic contractility and then low isotropic contractility. And then this we test by continuously doing laser cut on the band as the band is growing. So this allows us to probe the amount of contraction as a function of the nematic order directly. And again, this experiment I've told is extremely annoying to do because it's dependent on luck and you have to wait there for ages and then hope that you cut at the right time. But thanks to my collaborators, all this is done. And then you can see in this from here that indeed the contraction that we observe in the band is positively correlated with the nematic order, which is what we predicted. Okay, so what we learned from the active gel theory is firstly, it's similar to complete cell division in that we require very different contractility between this cable and then the rest of the cortex. But at the same time, it's also different from complete cell division in that we now get this depletion zone where you barely have any actin just because we have this flow that's away from the nematic band, that's away from the actin cable that creates this depletion zone. And then we want to ask, how does this affect the shape of the cortex? Like, how does this tell us about how this like partial cell division is achieved? So to see that, this is a membrane label, so we can see where the membrane is. This just helps us track how the cell is dividing. So you can see there are actually two kind of distinct phases. If we wait a little bit. So in the first phase, we can see this is indentation is happening. But in the second phase, actually, we see a new membrane that's just formed between where the cell was going to divide, and then it actually becomes a little bit less indented. So that's what I just said, but in a nice chemograph. So this is in phase 1. So this is just looking at the membrane marker along this line. In phase 1, we just see kind of increased indentation. And then in phase 2, we see that by formation of more membrane, it's able to progress through the cell division further while putting this point up. So to describe this, we could put active gel theory on a deformable membrane and then look at the very complicated, mesoscopic interactions between the membrane and the gel and the shape, etc., which was actually done in this paper. But another very nice thing that they showed in this paper is that you could also just write a microscopic mechanical theory of constriction while just treating all of this as a uniform material and essentially arrive at the same conclusion. So which is what I did. So this is the part where there's a little bit of false advertising because I said I was going to talk about active gel theory and now I'm not. But why do active gel theory on a deformable membrane when you don't have to? So the microscopic mechanical theory of phase 1 just involves writing down the total elastic energy of this entire system. To do that, so we just need to kind of count all the energy we have in the system. So the first part of the energy is the energy of the contractile cable, and the second part is the energy of the cortex. So the energy of the contractile cable is just the length of the cable times the width of the cable, and then times the surface energy of the cable minus the surface energy of the cortex. And then similarly, the energy of the cortex is just the total area of the cortex times the surface tension of the cortex. So all very simple. And then all the terms that's outlined in green, they're sort of inherent properties of the cable and the cortex that doesn't change over time. Whereas the rest, like the length of the cable and then the area of the rest of the cortex, we can obtain from geometry and volume conservations. So there are basically functions of this height. This is this h1, this height h1 that is just like the height of the cable relative to the bottom of the cell. So all in all, we can actually then simplify this energy as a function of H, and then it turns out to only depend on one parameter, which is kappa, which sort of tells you the contraction in the cable compared to the rest of the cortex. So kappa is equal to the width of the cable divided by the radius of the cell before the cleavage, and then times the difference in contraction in the cable versus the rest of the cortex. So this is a dimensionless parameter. And then when we plotted this energy for different values of kappa, we see that for when kappa is extremely small, then the cell doesn't do any cell division. The cell is happiest in this state when, because without any contractility, there's no reason for the cells to divide. And then as you crank up this contractility, then you can see that the minimum of the energy moves from when h1 is equal to r to when h1 is equal to 0. And then h1 equals 0 means when you have completed this cell division. And then in fact, if you then plot the minimum of this energy landscape as a function of kappa, you can see that it undergoes a continuous phase transition. Like that. So this is h, and then there's a kappa. You can see for small h is 1, and then as you crank up h, it undergoes a second order phase transition to when h is equal to 0. And then we compare this with the same theory for complete cell division. So it's kind of the same thing. We have exactly the same dimensionless parameter kappa, except the only difference is that rather than having this second order phase transition, we have a first order phase transition over here, where as you tune over values of kappa, we abruptly go from something that's like this to a complete cell division over there. So like this. And then we think the reason this is happening is that if you imagine the constriction that's in the second case, the ring is shrinking like this, right? Whereas in the first case, this contractile cable is going down sort of more like that. So initially it's a hemisphere and then it's approaching a straight line. Whereas in the second case, you have a ring that's turning into a smaller ring and then it's vanishing. And we think is that this difference in geometry, this is much more sudden, and then towards the end, constriction actually becomes easier. Whereas this is constriction does not get easier as you approaches the end of the cytokinesis. Okay, like that. And then we look at phase 2. So we could basically use exactly the same argument, except we have the energy of the cable, and then we have the energy of the cortex, and then we just need to add the energy of the contractile. We just need to add the energy of the new kind of membrane contact zone that we have over here, which is this term. So exactly the same as before. All these terms are inherent properties of the cable, the cortex, and the contact zone, whereas the rest we can obtain from geometry and volume conservations, and then they're all functions of h1, which is the height, which is what we had before, which is the height of the top point of the contact zone, and then h2, which is the point of the lower point of the contact zone. And then it turns out we only have 2 dimensionless parameters. So the first one is kappa, which is the same as before, and then the second one is nu, which tells you how energetically favorable it is to make the new membrane contact area. And then with that, we have our energy landscape in both H1 and H2. So let's just unpack this diagram a little bit. So when both H1 and H2 is equal to 1, then there's no constrictions happening. When H1 is equal to 1, when H2 is equal to 0, that means the top point, nothing is happening, and the bottom point, you form the entire membrane contact that just divides the 2 cells. And then you can see here, both in this cases and in this cases, you've completed the cytokinesis. So the cells wants to be kind of at the bottom line. And then you also have somewhere in between where the H1 is a higher point than H2, and this is why this region is the unphysical. This is the unphysical parameter region. This is the unphysical region in this energy landscape, as you can see over here. So all these 3 panels are showing that if we keep the same value of kappa, and then we just increase this nu. So remember, kappa tells us about the contractility of the cable, whereas nu tells how preferable it is to make new membranes. So if you just make it more and more preferable to make new membranes, then if nu is equal to 1, which means membranes is as expensive as the rest of the cortex, then you only do phase one. But if you make membrane a little bit cheaper, then you can see this point deviates from here and then is actually able to constrict a little bit further. And then similar way over here is able to constrict a lot further. And then one interesting thing is that by constricting further in H2, it actually increases H1, which is consistent with what we observe in experiments, where it's kind of making this membrane contact zone to divide further, and meanwhile the top point goes up a little bit. And then again, we did parameter estimation, we use experimental data from about 10 embryos, and then we can see it located us just by looking at the final value. So basically looking at we fit the minimum of this energy landscape to what we observed as H1 and H2 in experiments. We were able to kind of find these two best fit values, and we can see with this set of best fit parameters, we see that the kind of the basin of the energy landscape actually fits quite well with what we see in experiments. And then our collaborator did a very fun experiments where they put the embryo in calcium-free medium. And then what this does is that it stops the membrane contact from being able to form. And as you can see here, there's no contact. Am I over time? Yeah. 1 minute. Okay. Okay. All right. So by our theory, since there's no membrane contact, right? Then rather than arriving at the global energy minimum, it should only be able to reach the energy minimum of phase one, which is over here. So this is what we predicted. We take the same value of kappa and we put it into our phase one energy landscape. And then this is what we predict the calcium-free median should give us. And it matches surprisingly well with the experiments, which we're very happy about. Okay. So in conclusion, we wrote sort of theory at two different length scales. At the mesoscopic scale, we wrote active gel theory by kind of taking the minimal approach and then adding the ingredients as needed. We were able to arrive at a theory that reasonably described the flow field that's observed, as well as the density profile and the depletion zone. And then to look at the geometry of the constriction, we wrote a macroscopic theory that treats kind of all components as uniform, and then we arrived at the energy landscape that, again, recently described phase one, phase two. So I would like to thank my collaborators, Xin and CP, and then I would like to thank my group and all the people that gave me money in the past. So thank you very much.
Thank you. Are they a very quick comment or?
Yep.
Take one and then.
Yes, there's an advection by the flow field.
So the DDT has the advection terms?
Yes.
The big DDT?
Yeah, the big DDT. It's the material derivative.
Yeah, okay.
Yes, the flow field is considered compressible. I mean, because we are integrating over the third dimension, right? But I mean, also there's also the acting turnover rate and stuff. Yeah.
In your mesoscopic theory, you said that the two types of acting fields are coupled through a shared actin bath.
Mm-hmm.
How is that incorporated in the theory? The target densities?
Yes,
or
yes. So this is, if you allow me to go very quickly through my slides. So actually what we did is that firstly, we wrote a theory where they could both, well, we firstly wrote down the chemical equations exactly as this is. And then we realized if we just say the monomer density is large, then effectively what happens is that both of them have their own target densities. And then we make this target density vary in time such that the active actin is only seen in the band region, whereas the passive actin is seen in the rest of the cortex. Does that?
So practically they are not coupled?
They are, I mean, they're coupled through these two. We make these two add up to one.
Oh.
So we just assume that the transition rate is fast enough that it doesn't really matter. I mean, I also wrote a version of theory we wrote in like explicit transitions from this to the monomers and then the monomers back to this. And it basically does the same thing.